we are going live.
Is it okay to start now, Barbara?
Yes, okay, now we can start. And so welcome to everybody. I'm Barbara De Ruggeri, project manager of the EIT Food Hub in Italy. Later on at the end of the workshop, I will show you a little bit what EIT Food is for the agro-food system in Europe, and which are the opportunity for all of you students and entrepreneurship in the framework of the EIT Food. We are really happy to host this workshop today because basically all the applied biotechnologies for underutilized byproducts are exactly activities that fit into the innovation focus areas of EIT Food. Because we promote circular economies, a reduction in resource on land and water use. So all the results that will be presented today from the project FUNBREW are really important because can transform and innovate all the process and production in the European agro-food business. So now I'll pass the word to Rossana Coda that will make a brief introduction, and then we'll manage the first session.
Okay, so I will now share my screen. All right, so good morning or good afternoon, depending on where you are, and thank you very much for being here. I will just give a brief introduction, coming straight to the point on why we are focusing today on brewer's spent grain. It's probably all of you know, brewer's spent grain is the insoluble residue from the beer brewing process, and it represents a very abundant byproduct in the beer industry, which is also produced in huge amount worldwide, about 39 million tons per year, of which about 4 million tons only in EU. So brewer's spent grain basically consists of the husks of the barley grain that remain after the wort is filtered, but sometimes there can be also other types of cereals. It depends very much on the beer making process and the type of beer that is produced. Generally, we could say that brewer's spent grain is a lignocellulosic biomass. And in fact, if you look at the composition, this is an average composition that we can have for brewer's spent grain. It mostly is constituted by fibers of the lignin type or cellulose or hemicellulose, but it also contains a fair amount of protein and lipid. So brewer's spent grain has a huge potential as food ingredient, but because of its properties and its quality, it also creates several challenges if it is used as food ingredient. And indeed, the current main destination is for animal feed, and it has a very low market, if it has at all a market, if there is at all a market for this product. However, because of its very good nutritional composition, if used for human consumption, brewer's spent grain could contribute to healthy diets. It is rich in fiber, after all, and other bioactive compounds, and indeed could contribute to reduce food waste and improve what is the current European strategy of circular economy. So the question is, how can we use more of the brewer's spent grain in food? And this is where the FUNBREW project came into play. So our goals were to establish bioprocessing technology, namely fermentation or enzyme-assisted fermentation, to improve brewer's spent grain as food ingredient, to understand how the changes induced by these treatments would affect the characteristics of brewer's spent grain, and to evaluate the quality of food containing our bioprocessed brewer's spent grain. So the project consisted of the work of 4 research partners: University of Helsinki in Finland, the University of Bari and Free University of Bozen in Italy, and Rice Institute in Sweden. We also were helped by several industrial partners that contributed their spent grain, or they helped us with their biotechnology. So I want to just briefly introduce what you will be listening to today. We have divided this workshop into 2 sessions. One first session will mostly focus on the functional and nutritional improvement that we can obtain through fermentation and to tailored bioprocessing of the brewer's spent grain. And the second part will focus mostly on what happens when we have used bioprocessed brewer's spent grain as ingredient in pasta, breakfast cereal, and bread. So we will have a Q&A session at the end, but hopefully if we have time, also in the meanwhile, we can do that as well. And without any further ado, I would like to introduce the first speaker of today, which is Carlo Rizzello. Carlo, are you here? I will stop sharing my screen now.
Hi, Rossana. Thank you. I'm here and I'm trying to share my presentation now. Okay. Thank you, and thank you all for the participation at this workshop. In my presentation, I'll present some of the advantages of the bioprocessing applied to the agri-food side streams. As you know, the side stream can be produced at any stage of the food supply chain, from production to processing, distribution, and consumption. Since side streams includes byproducts, surplus, defective, expired products, and a huge amount of side stream are currently produced from any food supply chain. In many cases, these side streams are still edible, but regardless of this, the major part are discarded and used for feed and compost production, and only in small part for the production of biogas and bioethanol. Nevertheless, the side streams still contains nutritional compounds of nutritional interest. In many cases, they are rich in dietary fibers, proteins with high biological value, and also bioactive compounds like, for example, phenolics with antioxidant activity. For this reason, the downgrade of these side streams is a huge economic loss for the sector, and also an environmental issue due to the very high organic load in the environment. The side stream, as I told before, can be different. For example, we have the fruit and vegetable post-harvest loss, the unsold product, the pomace from the apple and tomatoes, for example. Also, cereal industry produce a lot of side streams. For example, the milling byproducts, the surplus and wasted bread, the brewer's spent grain, but all the food sectors, for example, the dairy sectors that produce the whey of the grape pomace from the enological sector, are interested in this, are involved in this huge production of side streams. The bioprocessing is a tool to recycle, reuse, and valorize these side streams through the use of microorganisms like bacteria, yeast, and filamentous fungi, and their enzymes. Obviously, bioprocessing also includes technological and physical treatments. And the aim of the bioprocessing, the aims of bioprocessing can be very different. The side stream, for example, can be used and optimized as substrate for the microbial growth. In this case, we can recover the microbial biomasses, for example, for the production of viable probiotics or for the cultivation of microbial starters to be used in the fermented food production, or also microorganisms that can be used for the biocontrol of plant pathogens or in biopreservation strategies, for example, to inhibit the growth of spoilage microorganisms in food. In another case, we can also recover metabolites from the microbial from the microorganisms that grow into the side streams. There are a lot of molecules of industrial interest, for example, bioplastic, biofungicides, and compounds with pharmaceutical and cosmetic uses. But we also have the possibility to produce food ingredients like free amino acids or organic acid and different functional compounds synthesized by microorganisms, but also converted by microorganisms from inactive forms that can be present into the matrix. These functional compounds can be used to fortify the functional foods or also to produce food supplement. But there is also another side of the bioprocessing of the side streams. Side streams can be converted entirely in food ingredients. Side streams are characterized often by poor technological and sensory properties, and often also by the presence of anti-nutritional factors that can limit the large-scale application as ingredients. For this reason, it is very important for the academic and industrial research to develop a proper bioprocess protocol that must include different considerations for different stage of production. First, the design of a quality control plan of the side streams, for example, the contamination by microorganisms and mycotoxins. Then it is important to define the pre-process condition, for example, thermal treatments, milling, homogenization, dehydration. An important point is the selection of proper starter microorganisms and enzymes, and the selection and the setup of the protocol with all the fermentation or bioprocessing parameters. Then it's also important to define the stabilization conditions of the bioprocessed biomass before the use as food ingredient. Among the different microorganisms that can be used for the bioprocessing, lactic acid bacteria are the most studied and the most employed, probably thanks to the long traditional use in food production. Lactic acid bacteria can contribute to the improvement of the technological and sensory properties of different side streams, but they also have important role in the improvement of the nutritional and functional properties. Lactic acid bacteria, for example, can increase the protein digestibility thanks to the proteolytic activity occurring during fermentation. They can also decrease the glycemic index because acidification promotes the formation of resistant starch. Lactic acid bacteria can degrade different anti-nutritional compounds if properly selected for specific metabolic activities, and can increase the bioavailability of soluble fibers, minerals, antioxidant compounds, and moreover can enrich the matrix with functional compounds, bioactive compounds that can be synthesized by these microorganisms. In this way, the processed side stream can be used as ingredients for the fortification of staple foods like pasta or bread or fermented beverages, or for the formulation of novel and innovative functional foods, combining the properties of the side streams, for example, the dietary fiber content and the advantages due to the fermentation. Just a few examples of some bioprocesses in which our research groups have been involved in the last year. For example, we worked on milling byproducts, the cereal bran and the germ, by select lactic acid bacteria able to stabilize the lipase activity and to prevent the rancidity of the cereal germ. We select lactic acid bacteria able to degrade phytic acid, that is the main anti-nutritional compounds contained in bran and germ. And in this way, the milling byproducts can be reused as a source of dietary fiber and included in new formulation of baked goods and other foods. We also develop some bioprocess on grape pomace by using it as substrate for the cultivation of probiotics bifidobacteria. And we also use the grape must that in many geographical regions, like the south of the Italy, is a surplus. And we use must for the cultivation of a selected lactic acid bacteria able to produce gamma-aminobutyric acid, that is an important functional compound that can be used in food application, but also in cosmetic application. We are involved in a European project on the valorization of wasted bread. In this case, we are working and we worked on the definition of protocols to reuse bread into the bakery as an ingredient enriched by fermentation in antifungal compounds, gamma-aminobutyric acid, and also compounds that can have technological advantages like dextrans, that is a type of exopolysaccharides synthesized by lactic acid bacteria. We also used wasted bread as substrate for the cultivation of yeast and lactic acid bacteria to be used as starter for baked goods fermentation in a point of view of a circular economy into the food industry. The last example is an application to the whey, that is another important side stream that is still rich in lactose. We optimize a protocol to cultivate a bacteria called Alopherax mediterraneae that is able to produce a bioplastic PHB, a compound that can be used as alternative of petroleum-derived plastic for producing, for example, packaging for food purposes. And yes, I'd like to thank my research team in Bari and my colleagues in University of Helsinki and University of Bozen that collaborate to FUNBREW and the other project I showed to you in this brief presentation. Thank you all.
All right, Carlo, thank you. And I would like now to ask Marta to prepare her presentation and share her screen.
Hi, Rosanna. So
Hi, Marta.
I will share my screen.
Yes, thank you.
Can you see it?
Yes.
Can you see if I change the slide?
Definitely, you can
Okay,
start.
Thank you. So good morning, afternoon to everyone. So as Rosanna pointed out, and also Professor Rizzello, I will speak about the starter selection to exploit VSG as a food ingredient through lactic acid fermentation. So the work we did in Bolzano was focused on screening lactic acid bacteria in order to select the most performing ones, which had the metabolic traits more appropriate to ferment the VSG. Not all bacteria or all species are suitable to perform a food fermentation, and even inside lactic acid bacteria, even inside the same species, like in lactobacilli, there are several differences even among the same species. So we have to assess properly the capabilities of these strains for our substrate to be fermented. For this reason, our group focused on the starter selection, not based in in the genomic capacity, for instance, but based mostly on the metabolic traits. What they can actually do and how well they do it. So metabolic traits rely on the genetic information which is encoded in the genome. So the genomic portfolio this strain possesses, but also relies on the environmental pressure they are exposed. For instance, in our case, the BSG ecosystem. So in order to do so, we know, as it was pointed out, that the raw BSG doesn't have good technological and sensorial properties, but has kind of good nutritional properties. As Rosanna pointed out in her first presentation, BSG is mainly constituted by hemicelluloses, celluloses, and lignin. So it's a quite harsh environment for lactic acid bacteria that they have to overcome several challenges. So we will focus on which bacteria we will use in order to perform this lactic acid fermentation. So we selected 4 strain candidates to be further screened based on previous studies. So 3 of these strains were belonging to Lactobacillus plantarum species that is widely used in food fermentations because of their metabolic flexibility in different environments. And also we selected Leuconostoc mesenteroides to be further screened because of a previous study and their capacity to produce dextrans, which can improve the technological properties of foodstuff. So our approach was based generally to see how these 4 strains would behave in a standard environment like is MRS, and then how they would behave in the water-soluble extract of a BSG coming from Italy. In order to assess this, we use the high throughput phenomics approach, which is assessing in a kinetic way, so over time, how different substrates are consumed. It is not a growth assay, it detects directly the metabolism which is happening due to the consumption of different substrates. We mainly focus on carbon sources, and we screen over almost 200 substrates, and we saw how these strains were consuming this substrate if they were previously adapted to an MRS standard media and compared to a BSG-like media. This was the main technique used, and this technique generates a large amount of data. So we also have in our group a pipeline to process all of this data. Then we double check how during fermentation of this water-soluble extract, lactic acid was produced, and also some organic acids that are important for lactic acid bacteria. And then based on phenomics results, we check the gene expression for metabolic pathways that are related to this substrate consumption. So we saw that, as you can see here, that Lactobacillus plantarum, the 3 strains, group their phenotypes according to the media they were cultured. And this suggests that the environment had a more important effect rather the genotype, that is the different strains. So, but also Leuconostoc pseudomesenteroides show a very distinctive metabolic profile, but we could see that there was a change between MRS and the BSG soluble extract. So this was the area under the curve of the metabolic curve for the substrates that show a consumption during the experiment. So we can say that because of BSG-like media, there was a metabolic switch from MRS standard condition to this new ecosystem. And the fact of plantarum, because the phenotype also reflects the species and the strain genomic profiles, we see that plantarum, even if they were different strains, they grouped together in their phenotype profiles depending on the media they were cultured. This is because plantarum is a nomadic lactobacilli, and nomadic lactobacilli have a very wide range of metabolic activities that are preserved among strains. There are no strains very adapted to certain environments, but they are more like they can cope with different environmental pressures using kind of the same metabolic strategies in an overall picture. But then we want to see more in detail how this substrate consumption was performed over time, and if we could see differences among strains. So here, for instance, you have the metabolic curves for selected substrates that we found they were interesting for BSG, and also we saw that there was a lot of change in the activity when the strains were moved from MRS to BSG. And as you can see in the dashed lines, this is the metabolic curves for the 3 strains cultured in MRS. And generally, you can see that, for instance, for galactose, there is a big increase of the activity when the strains are moved to the BSG. You see the metabolic curves are rising before, and the shape may change. And this is what we did. We assessed over different time points how this effect was taking place and how it was evolving during time, and also how significant it was respect to the control condition per time point of the experiment. So at the end, we can see okay, so you have the three strains of plantarum cultured in MRS, and these are the different substrates, and how from MRS to BSG, how important the metabolic switch was. And we see that almost for all substrates, when the strain PU1 was cultured in BSG, there were a lot of significant effects caused by BSG ecosystem into the metabolic performance for a wide range of substrates. So for this reason, we selected PU1 as the Lactobacillus plantarum strain that could cope better and shift its metabolism towards a more efficient metabolic performance. Then we also check how the organic acid profile was evolving during time. So as you may know, in MRS, there is only one carbon source, which is glucose. And we see that the production of lactic acid, because of the presence of glucose, was kind of linear and only show one exponential phase during time. And kind of the same for lactic acid. But when we culture the 3 plantarum strains, but also the Leuconostoc pseudomesenteroides in BSG, we saw that there was a first production of lactic acid, then there was a kind of plateau phase, like a lag phase, and then the strains continued to produce lactic acid. And kind of the same profile was observed for acetic acid. And this suggests that because BSG is not a defined medium like MRS, we may have, for sure, we have different carbon sources. So first they consume one carbon source and then they stop, they change their metabolism and then continue producing lactic acid because they are consuming a different one. We also check, according to the phenotype results, the expression, like the intermediate phase of the cellular process before the phenotype, we check the expression of different genes related to the substrates that we observe were changing more in the phenotype analysis. So what we saw was that, in fact, the expression of different genes was for plantarum strain dependent. PU1 preferred, for instance, to consume arabinose in the first metabolic phase, while the second strain preferred to consume these arabinoids in the second metabolic phase. Also, we see that PU1 had an increase of expression for galactose and galactans related genes, while the second strain did not. And also we saw that cellobiose related enzyme related genes were more expressed in PU1, which was the most performing strain according to the phenotypic results. For leuconostoc, we also saw that there was an increase of sucrose consumption and cellobiose and xylose, mostly during the second phase. So this study allows us to perform a starter selection based on the final output of the cellular processes, which is the phenotype, the metabolic capabilities of the strains. And we saw also that even being the same species and following the same metabolic strategies, the degree of adaptation of the different strains, it varies among them. We saw that in fact PU1 was the highest metabolic performing strain, and it showed the highest capacity to adapt to BSG ecosystem. We also saw that mesenteroides, pseudomesenteroides and plantarum degraded substrates that were associated to fibers which are contained in BSG, like some hemicelluloses and cellobiose building blocks. Thank you very much for your attention, and I give the word to Rosanna.
Thank you, Marta, for this presentation. And now I will invite Michela to share her screen. Good morning.
Good morning.
We are right on time, so we can start.
Okay, so good morning, everyone. And this is it. So as Rosanna said, 39 million tons is spent grain generated to take action on the matter. This process is not sustainable. Currently, the majority of spent grain produced is used as low-value animal feed, whereas a little part of it is used to produce biogas or dispose of as landfill. And so imagine not only the cost of disposing of such huge amount of spent grain, but the environmental impact of the product, because the 80% of it is made of water. Yet if you look closely, you'll see that the remaining 20% is made of fibers, also rich in
Michela, I think your connection is a little bit unstable and you are frozen now.
Rosanna, probably would be better if you share the presentation so that connection is not so heavy for Michela.
Okay, I can. I'm Carlo. I can share the presentation of Michela.
Yes, sorry, Carlo. Yes.
Yes.
But we need Michela to stop his sharing, her sharing.
I think she did.
Okay.
She's out anyway.
Can you hear me? I'm back.
Yes.
Yes, we can.
Carlo will share the presentation, so your connection will not be so heavy. Okay.
Okay, thank you.
Let me know if you are.
I was at slide number 4.
Yes.
Okay, maybe I can.
Yes, let's restart.
Okay.
Okay, thank you.
Okay, so as I was saying, the majority of spent grain is used as low-value animal feed, whereas a little part of it is used to produce biogas or dispose of the landfill. So imagine not only the cost of disposing of such huge amount of spent grain, but also the environmental impact of moving this much highly perishable product, because 30 or 80% of it, 80% of it is made of water, and if you look closely, you'll see its potential, since the remaining 20% is made of fibers and proteins, and it's also rich in phenolic compounds. Slide. And this begs the question, why isn't brewer's spent grain used as food ingredient? Well, its use is limited by its complex structure, which has consequent poor technological properties on food that contains, and requires treatments to reduce the negative impact on both texture and sensory properties of the final product. Slide, please. Thank you. So this is basically the aim of the FUNBREW project. We use fermentation and enzymatic treatment to improve the nutritional and technological properties of spent grain. On one side, we work on 2 fronts. On one side, at the University of Bari, we used fermentation coupled with enzymatic treatment to enhance the antioxidant properties of spent grain, whereas at the University of Helsinki, spent grain was used as substrate to synthesize natural structure-forming compounds known as dextrans, which improve the technological properties of spent grain. So let's take a closer look to what we've achieved. Slide, please. After a screening of 33 lactic acid bacteria, we selected a protocol involving a commercial silanase and fermentation, based on the ability of the strains to improve the antioxidant the antioxidant properties in vitro. Slide, please. Bioprocessed spent grain had scavenging activity against several radicals, and also showed lipid peroxidation, inhibition of lipid peroxidation comparable to that of antioxidant used as reference. However, since in vitro assays can only be considered predictive tools of what happens in vivo, ex vivo assay offered the right compromise to better understand which are the functional properties of food. And we used spent grain extracts and tested them on keratinocyte cell line, and we observed a protective effect of oxidative induced stress on these cells, especially in the extract obtained by processed spent grain. In this the effect was comparable to that of tocopherol. Slide, please. And these improvements were obtained thanks to the enzyme which freed phenolic compound bounded to lignocellulosic material, and also to lactic acid bacteria, which metabolize phenolic acid and obtaining more active forms. On the other hand, lactic acid bacteria proteolytic system really enabled the release of small bioactive peptides, which are small sequences of the native protein encrypted in the protein, in this case, barley's and maize protein, and showing common feature of antioxidant peptides. Slide, please. Now, as I told you before, being rich in fibers, it's when added to food, especially if in high quantities, spent grain compromises the structure of the final product. However, there are some lactic acid bacteria capable of producing substances known as isopolysaccharides, which, among the other properties, have water binding ability, and they act as hydrocolloids and stabilize the liquid dough interface and prevent the collapse of the structure. Having these properties make sure that we don't use commercial hydrocolloids, making sure that the products can be labeled as clean labeled. Slide, please. So after a screening of several lactic acid bacteria, a fermentation protocol for dextran synthesis was used, and spent grain added of sucrose was used as substrate. Slide, please. The selected strains were able to generate relevant increases in the viscosity. In fact, after 24 hours of fermentation, dextran content was approximately 1%. Also, oligosaccharides were formed, and this is also this also can be a functional property, since oligosaccharides can act as prebiotics. And spent grain enriched in dextran were then used by Rossana to make bread, was used as ingredient in bread making, and later Rossana and Andrea will talk about it. So to conclude, by increasing the shelf life, the functionality and the technological potential of the spent grain, the protocol set up in the FUNBREW project can help improve the sustainability in the agricultural food sector, valorize side streams and waste from the food processing industry, and contemplating their utilization within the food chain. So remember, bioprocessing with lactic acid bacteria can get us a step closer.
I think I have lost the last sentence, but it was really the last sentence. So first of all, thank you, Michela. Indeed, we will see more about the dextran production in bread in the next session. So I suggest now that you, Michela, especially take a breath, because you will be the next presenter. So we will have 2 minutes now of, 1 minute of a very short break. The time that Carlo can prepare the next presentation of Michela, and we can then start straight away the next session. Seems like the connection is a little bit dancing today.
Okay, Rossana, I'm ready with the second presentation of Michela. I don't know if it's better to change the order of the presentation and introduce Lily. The microphone, Rossana.
Oh, sorry. Yes, of course we can do that. If Lilia is with us and she agrees to start, we'll give Michela a little bit of time to check her connection or connect again.
Sure, absolutely.
Just
just a bit taken aback, that's all. Here we go. Is that coming through to you yet?
Yes.
Super, thank you. Full screen, please. There we go. All right. Yeah, my name is Lily. I'm presenting this work that we've done here at the Research Institute.
Sorry, Didi, you have to modify the display setting and invert the presentation.
Thank you.
Don't worry.
That's there's a bit of
No, just up. You say display setting, just in the bar.
In the bar.
Up is the display setting.
Sorry, I'm looking.
Okay.
This is not going well for me. I'm sorry.
Don't worry.
I'm not showing anything right now, am I? No. Okay.
Share again.
Yes, ma'am. All the technological jinxes that you can have at once. It's, yeah. At least I'm coming through. I'm glad for that.
Hot.
At least.
Everybody said it's a very hot day.
I wish I could actually. I wish I could blame the heat, but to be fair, it's only 26 degrees here in Sweden. Well, how's Michaela doing? Because maybe
just share the screen from Zoom. The green button, as you did before.
Lily, your mic is off, and if you prefer, I can share your presentation if you prefer.
I guess we'll have to do that, Carlo. Yes, thank you.
Okay.
I'm sorry about the mic as well. I don't know how that happened.
No problem.
Okay, I think now it's okay.
Yes, we can see that.
Lily, you can start. I can't hear you.
We can't hear you.
Lily, check your mic, please.
Unmute there. Thank you. Thank you very much,
Okay.
Carlo. Thank you for the save. Okay, sorry about the very embarrassing introduction. I work at the Research Institutes for Sweden, so you'd think I'd be able to handle Zoom, but we work a lot with Teams, so I'm very embarrassed. I'm sorry about this. So here at RISE, we've been working with extruded breakfast cereals, and I've had the pleasure of working with my colleagues Annika and Michaela, and they've been able to guide me in this process. So here at RISE, I'm one of the in-house extrusion experts. And of course, I could sit in the lab and develop breakfast cereals that taste absolutely horrible. But Michaela has been my my partner that has stopped that from happening by bringing in sensory panels for evaluation. And Annika also helps me with the texture of the product. Next slide, please, Carlo. So I'll give you a short introduction into extrusion. Perhaps not everybody's familiar with that. And then I'll go on to tell you a little bit about what brewer spent grain at 30% in breakfast cereal does to that breakfast cereal and how the consumers perceive that, and a little bit about the texture microscopy. So I'm very proud to announce that we've managed to put as much as 30% brewer spent grain into our breakfast cereal, which I think in itself is quite amazing, but it also has a serious impact on the texture of the product. So extrusion, in its most simple form, is what you see here on the side. My grandma, bless her, had something very similar to this in her kitchen. So it's basically a meat grinder. You can see that there's a big crank on the side as you turn this around and around. There's a screw in the center that will convey the material forward. So you could put meat in, you could put fruits in, you could even put a dough mixture in. So then as this is conveyed forward, it's brought to the whole plate you can see at the end, and there it's squeezed and formed. So in this case, if you were doing pasta, you would get spaghetti out. And of course, if you had mince or something, or meat, it would make a mince out of it. May I have the next slide, please, Carlo? Thank you. So up in the right-hand corner, you can see a schematic representation of extrusion cooking. So I'm taking this to the next step. I can do a little more than Grandma could do with her simple extruder. So what's happening here is as the material is conveyed forward, it is both sheared against the sides of the barrel. So this is very similar to what happens when you make bread and you knead the bread. So structures in the material are opened up, and particularly here it's starch that I'm working with. And then as this mass is transported forward, the heat also helps for the starch to melt and form a network. And then when it reaches the nozzle, it's then pressed into a shape, and that's what you can see down at the bottom left corner. I can promise you that that ribbon, that dark brown ribbon, that is actually the 30% brewer's spent grain, but it's perhaps not that exciting as it is now. So if you just took that and dried it, it would have the consistency of a dog chewing toy. It would be really, really horrible. So instead, what you want to have is a puffed product. In extruder language, that's called expansion. And it's basically the same principle as you have when you make popcorn. So it's just that there's water entrapped inside the maize kernel, or in this case, in this ribbon of starchy product. And then when you make the water evaporate quickly, as you do when you put your maize kernel in oil, then that water evaporates and it leaves behind little pores. So it gives this fluffy structure. On the bottom right-hand side, you can see this amazing image that Tova and Annika managed to capture in the lab. So the purplish color is the starch, and down at the bottom you can see whole cells that contain small starch granules. Sorry, I don't have the pointer to show you right now. You can see down at the bottom intact starch granules, and then as you move up through the picture, you can see a cell that's starting to swell, where you can still see there's distinct boundaries between the starch granules. And then up at the top, you have a starch network, and that is what keeps together the product so that you can get this nice puffed structure. So that's enough on extrusion. If I could have the next slide, please, Carlo. I'll go directly on to. Oh, right. If you feel inspired, you could also do this at home. It's a very simple recipe. It's just a little rye flour, some starch to make sure that you actually have that nice starch network. And then here's the 30% brewer's spent grain and a little bit of table salt. And of course, this is not just invented straight out of the air. It came about by, we brought in a focus group. So that's a bunch of consumers that we just presented a bunch of different types of cereal that are on the market. And we asked them, hey guys, what do you like and why? And our consumer group chose the rye puff that was on the market as their favorite. This does, of course, reflect to a very large degree the consumers that you ask. So if you had asked a bunch of kids that were 7 years old, they would probably chosen something with a lot of color. But our consumers were mature females and they wanted something healthy. And this rye puff is what they then expected. They also guided us how much salt we should have in there. And to my great amazement, they were the ones that chose that we should have 30% brewer's spent grain. If we have the next slide, please, Carlo. Yeah, there is quite an impact when you add that much brewer's spent grain to the breakfast cereal. So at the top, you can see these nice light colored puffs. That is the rye base. It has no added brewer's spent grain. And I was seriously expecting our consumer groups to say, we like this one best. But we also gave them 15% brewer's spent grain and 30% brewer's spent grain. You can see them at the bottom. They're a bit darker, and you can see they're a lot more flat, so they don't have this puffed structure. But they like that one best. Maybe why, we can get back to. I'll just tell you a little bit more about what else. You can obviously see this difference between the puffed and the color as well. They could taste these and they could see that the brewer's spent grain made the sweetness decrease a little bit, and there was a little bit more bitterness, but not so much that they disliked the breakfast cereal. We also presented them with brewer's spent grain from different breweries, and it was quite clear that the raw material has quite a large impact on the final breakfast cereal. So depending on which type of brewery the brewer's spent grain came from, they could distinguish. And then we also went on to compare fermented and unfermented brewer's spent grain. So we were using dextran producing microorganisms, as Michaela was just telling us. And it was quite interesting, I found, because it was a bit inconclusive results. So for Dugges, one of the breweries, that brewer's spent grain was perceived as better when it had been fermented, whereas for Senthon it was the other way around. So that was something I think is worth investigating further. May I have the next slide, please, Carlo? I've bored Carlo to sleep. Yeah, thank you. There we go. Up again in the right-hand corner, you can see the difference between the rye base that has no brewer's spent grain and the one that has 30% brewer's spent grain. So this is just to tell you that we also did some measurements with this instrument. It's basically just a piston that presses down on the product and it measures the force it takes to puncture through the product. And we could confirm that, yes, there are indeed less pores and the structure becomes more compact when you have 30% brewer's spent grain. This is entirely what we would expect. We've seen this in other extrusion projects, that as soon as you add a lot of dietary fiber, the structure collapses. And I think Michaela was saying something similar about bread also being more compact when it contains brewer's spent grain. However, there is a silver lining to this, and because there are less pores in the brewer's spent grain cereal, it also means that it doesn't absorb milk quite as quickly. So we had a student in the lab. She did these measurements also with milk, and she waited for 20 minutes, and I think that was as long as she could wait. She was very impatient, but she also said that 20 minutes is about the longest that somebody will sit with a bowl of cereal. And at that point, the breakfast cereal was still a bit crispy. So it was not this soggy mass which you otherwise have with many breakfast cereals. And then finally, may I have the next slide, please, Carlo? Thank you. We have some beautiful images made by Tova and Annika in the microscopy lab. So here they've used a marker to stain the Arabinos Islands. They sit in these structures. It's not the outer, it surrounds the starchy part of the grain. And those are the red bits that you can see. They look, I mean, you can see that they're cell shaped. And then the green stuff is, well, I can't really tell you exactly what it is. It was not intentionally stained. It's just autofluorescence. But I think both with the green and the red, you can see that when it's fermented, it's a little more fragmented, which is, I guess, also what you could expect to see then, that fermentation would increase the fragmentation. However, we didn't really see a big difference between fermenting with sugar or fermenting without sugar. So one would increase the dextran production and one would not do that.
Lily, you have 1 minute.
That's super, because I'm at the last slide, if you please take it.
Okay.
Yep. So that was my story on how we developed a breakfast cereal with 30% brewer's spent grain. We confirmed by several different types of measurement that the texture becomes a lot more compact with the brewer's spent grain. And we also noted that brewer's spent grain is not all the same, so it has an impact on the final material, depending on which type you use from the beginning. And it's not necessarily all bad that it becomes more compact, because that allows you to eat your breakfast cereal more slowly. Thank you very much. All right, and I, of course, have to thank all my beautiful collaborators, especially those in close collaboration here at RISE, but also our FUNBREW collaborators. And of course, this had not been possible without funding.
Thank you so much, Lily.
Thank you.
I don't know if Michaela is now
I'm
connected.
Here.
Yes.
I'm connected. Maybe it's better if I don't use my video.
Okay, and if you want, I can share your presentation now.
Okay. or maybe I can try. Oh.
Just one moment. Because now I have a lot of presentation of my desktop. And the presentation is ready. It's ready
Yes.
also for you. Okay. You can start.
Okay, thank you. So have you ever wondered why for the last decade fortified foods have been invading market shelf? Next. Health diseases associated with the diet, especially if rich in animal sources, are increasing, and studies have suggested that changing eating habits may help improve health. However, although animal products will continue to play a critical role in the global food system, this model is not sustainable anymore for both health and environment. Next, please. Which is why the Food and Agriculture Organization recommended increasing the intake of fibers and proteins from vegetable sources. And the easiest way of doing it is to fortify food, staple food like bread and pasta. Next, please. For this reason, FUNBREW aimed at not only recycling spent grain, but also making it a valuable functional ingredient for food application, among which pasta. Next. So spent grain bioprocessed according to the protocol I talked to you about before, the one that allows us to increase the antioxidant potential, was used to replace semolina at 15% to produce a fortified pasta. Next. According to the current European regulation, this pasta can be labeled as a high fiber and source of protein. And compared to native BSG, the use of bioprocessed spent grain led to higher protein digestibility and higher quality indexes, and as well as lower predicted glycemic index compared to semolina pasta. Next, please. But most of all, bioprocessing improved the technological properties of fortified pasta. In fact, the degradation of Arabic nucleoside structure resulted in the release of a component entrapped into the cellular compartment and a more homogeneous protein network. The extensive cell wall disruption contributed to the release of phenolic compound, which conferred antioxidant properties. And in fact, the digested pasta was observed. The digested pasta also had a protective effect towards induced oxidative stress in CACO2 cells. Next, please. And the fortified pasta showed a peculiar sensory profile with a scent of cereal, malt, and sourdough bread, and also a bitter taste among the main attributes. Yet markedly improved by the treatment, thus confirming the great potential of bioprocessed spent grain as health-promoting food ingredient. So to conclude, next, the underlying idea behind functional food is to reduce the prevalence of diet-related disease by modulating the consumption of high-value foods. Therefore, the incorporation of bioprocessed spent grain in food formulas can be a valuable option to provide compounds beneficial to the diet, revitalize stall markets, and fulfill consumer needs for healthier food, while foster cooperation among several players in the supply chain. Next, so a future where the cooperation of beer and pasta industries can contribute to a healthier and more sustainable diet is not that far away. And thank you, everyone. Thank you.
Thank you, Michela.
Thank you, Michela. Now I invite you, Rossana,
Yes.
for the last presentation of the session.
All right, thank you. So this is a shared presentation between me and Andrea. we will be talking about the use of brewer's spent grain in bakery. I will skip this slide because Michela has already introduced the problematic of using fibers, especially when the fiber content is very high. It is a problem, can create challenge, and this has been seen also in previous studies with brewer's spent grain when it was added to wheat bread. It did improve the nutritional profile, but caused smaller volume and denser structure. Always from previous studies, we saw that bioprocessing like enzymes and fermentation could improve the quality of bread in that case. So this process was about adding brewer's spent grain fermented containing dextran naturally synthesized in the way that it was already explained before. And the brewer's spent grain amount that we were able to add on a wet weight, it was around 33%. So if we look at the picture, we can see that the picture speaks for itself. When we see that there is the presence of dextran inside the bread has higher volume compared to the regular fermentation of brewer's spent grain or compared to no treatment at all of the brewer's spent grain. And indeed, we were able to see at least 13% increase of volume, reducing stalling rate and also a significantly softer bread compared to the other types. What is more, we also calculated based on the EU directive, and we saw that the nutritional profile of these breads was also advantageous because it could be labeled as source of fiber and source of protein. And coming to the sensory profile of the bread, we could see that generally, although fermented, the sour smell was really very minor in all the types of bread, and that indeed the brewer's spent grain bread, that is this IPS plus brown, had more homogeneity and indeed generally a slightly stronger odor intensity. And thinking about the mouthfeel and taste, we also noticed that the panel defined that the brewer's spent grain containing dextran produced a bread with higher moisture, more cohesive texture that was more appreciated in that sense. So dextran effect could be perceived also by the consumer panel. And now I leave the word to Andrea.
Thank you, Rossana, and thank you for your kind invitation to this event. I'm Andrea Minisci. I am R&D manager of Valle Fiorita, which is an innovative bakery company located in southern Italy, which produces bakery products like sandwich bread, flatbreads, pizza bases, and others, mainly for Italian grid distribution market, but also for other European countries. And Valle Fiorita is a leader in sandwich and bakery products market with the use of sourdough and wet flour. So we use a type 2 sourdough in liquid form for the production of all our bakery products. In fact, we have implemented in the last years a complete production and management system of type 2 sourdough, which allow us to take under control at every moment the main parameters of sourdough fermentation like pH and temperature. The sourdough is directly fed by our selected starter cultures that are prepared by our R&D team in the internal microbiological laboratory. In fact, we have implemented in the last years an internal laboratory in which our R&D team is very skilled in the propagation of starter cultures, but also in new product development activities. Valle Fiorita, next please. Rossana, if you can change the slide, okay. Valle Fiorita has become in the last years also involved in a circular economy project, and between them I'd like to highlight two projects which are still important for us now. In the first, our wasted bread has been converted into a substrate for the cultivation of starters for the food industry through a biotechnological process which has been patented in the last years. And in the other project, in the second, we have used two different kinds of milling byproducts like wet germ and bran, which have been converted through a fermentation process into an innovative bioingredient that can be used in bakery products, and Vale Fiorita uses it for a special line of bakery products with a low glycemic index, prebiotic activity, and source of proteins and fibers. Next slide, please. Also, the participation to FUNBREW project was very important for our company because we tried to use an underutilized byproduct like brewer's spent grains as a bakery ingredient. In fact, we prepared a special recipe in which the fermented brewer's spent grains were used through Helsinki University protocol in order to improve the production of dextrans, and were used in a special recipe with the other baking ingredients like wet flour, salt, yeast, extra virgin olive oil, and water. And so we made two different kinds of bakery products as sliced bread and focaccia bread. Next slide. These prototypes were very interesting from a nutritional point of view because they were high in fibers and a source of proteins, but they were very similar also to conventional whole grain bread in terms of textures and colors, as you can see in this table. Moreover, we organized an internal panel test to evaluate the difference between the conventional whole grain bread and the prototype bread derived from the use of brewer's spent grains. Next slide. Before the industrialization of finished product, there are some problems to solve, like the need of further industrial scale trials and taste optimization, and the need of an agreement of a supply chain between the brewers, the farmers, and the final factories that have to convert these byproducts into an added value product. And finally, also the implementation of a special equipment of wet milling for spent grains homogenization in order to improve the chewiness of finished product. Okay, at the end, I want to thank the entire team of FUNBREW project and also the R&D team of Vale Fiorita that have supported us during the duration of project. And thank you again for your attention, and I cede the floor to Rossana.
Thank you, Andrea. And I think with this we have concluded our presentations. So we are lucky because we have quite some time for the Q&A session. I will stop. So as Barbara said initially, please type your questions in the chat, and Carlo and I will moderate this one. I have, maybe you can start because I have one question, and I think that can be addressed to Michela. I think, Michela, you showed that there was some antioxidant activity on some skin cell line of the fermented brewer's spent grain. But does this mean that this type of fermented brewer's spent grain could have an effect also on the skin? Could that be used in some different ways?
Well, I think to know for sure, we should try. I mean, we need tests to make sure that this works. But And I'm not a skin expert, so but I don't see why not. I mean, if the idea is that the substances in brewer's spent grain, substances that were liberated to try through the by processing, are capable of increasing the antioxidant activity and block those substances that make the opposite, so radicals and whatever, why shouldn't be possible?
That's right. So there is another use that is cosmetics that might be studied, perhaps,
yes,
if the antioxidant activity is indeed so high. Then we have some question in the chat. There is a question from Heikki to Andrea. So the question is, what do you mean by type 2 sourdough?
Yes, instead of traditional sourdough, which is prepared in traditional bakers' factories, type 2 sourdough is produced under controlled fermentation cycle. So with this system, we can control at every moment some parameters of fermentation like pH and temperature, because our system is composed by special equipment, which are mainly fermenters, that activate a cycle of fermentation in an automatic way. So the difference between the two kinds of sourdough is mainly this.
And also, Heikki, if I can add something, because the protocol of fermentation has been developed in a research program with the University of Bari. In the company, selected lactic acid bacteria are inoculated into the liquid sourdough. And in the company, they have a protocol for propagation and for re-inoculum of the selected starter at some point with the check and monitoring of all the process. And the starters are only lactic acid bacteria. This is better for control, for monitoring the fermentation, especially if byproducts are used. Yeast is added in a second step of the production.
Thank you, Carlo. There are a couple more questions. We have a question from Angelo, and he's asking us, have you explored solid fermentation using fungi? No, we haven't. I can answer this question. I think that this project was our first deep dive into the marvelous challenges that brewer's spent grain poses us. So we only focused here on lactic acid bacteria fermentation for application in cereal-based products. So it's mostly a type of sourdough application that we developed, but I think the future entails a lot of different opportunities for brewer's spent grain and other byproducts indeed. Then there is a question from Chiara, and it's a question for Andrea. She says that she's interested in the panel test of the whole wheat bread versus the prototype of brewer's spent grain. Which one was preferred? What are the main differences? Can you tell us something, Andrea?
Yes, there were no strong differences between conventional whole grain bread and brewer's spent grains bread. They were very similar under some parameters like colors and chewiness or hardness. But we noted that for a chewiness point of view, the fermented brewer's spent grains bread has to be improved. In fact, the reason why we choose a wet milling equipment is for that, because we have to reduce the granulometry of brewer's spent grains in order to improve the chewiness of this byproduct inside the finished product, the bakery product.
If I may add something to...
Yes, Michela, please go ahead.
When we made the panel for the product, the panelists preferred the focaccia type, which is the flatbread product they produce in Valle Fiorita, and especially because of the elasticity, which is mainly because of the way they produce it, although it was the same dough. And also, of course, we also tried other products with lower quantity of brewer's spent grain, and those were much more appreciated, of course. But as Andrea said, just like they were mostly comparable to all wheat bread, the innovative products. That's it.
Thank you, Michela. I think I can say something about it because when we produced our bread prototypes at the University of Helsinki, we indeed do possess wet milling equipment. So we were able to reduce this fermented brewer's spent grain into quite a fine paste. So it did not really pose that type of challenge anymore. And indeed, when dextran was present in the bread, this was also perceived by the panelists because the bread texture was more homogeneous, it was softer, and I also had a personal preference because it was much sweeter and tasted rather good, I have to say. We have a new question.
Excuse me, Rossana, if I can add something.
Yes.
During another difference between this prototype and the conventional whole grain bread is that during the kneading phase of process, we noted also an improvement of hydratation of the dough, probably because there were a lot of dextrans inside the fermented brewer's spent grains. And so this effect is very important also for the preservation of finished product.
Yeah, I agree with what you said. We have some comments from Marie. She's saying that these are interesting results, and she wants to know what are the next steps for implementation and industrial valorization or next research steps. There are, of course, many interested companies, and how can we proceed? I will say very briefly that we will continue working with brewer's spent grain in different endeavors. Particularly at the University of Helsinki, we have a new collaborative projects that will explore a completely different food sector, that is beverage. And I can tell you for sure that since when we started the project almost 4 years ago, the number of scientific articles on brewer's spent grain on search engines like Google, it has increased a lot. So this tells me that research for food application on these byproducts is indeed going on. And concerning what would be the next step for industrial implementation, would you like Andrea to answer this question? What could be done in this sense?
Surely we need to make other industrial scale trials, because we worked only on pilot scale, and we need to optimize the taste, but also the industrial parameters of our equipment that are not suitable for this kind of process. So the aim of these industrial trials is this.
I agree. And thank you, Eiki and Luca, for being here, and everyone else that is still here. I think we are really right on time, and we might then probably leave the floor to Barbara, who wants to tell us about something about EIT Food Network, and then I will have the last conclusive remarks. So Barbara, the floor is yours.
Thank you. I'm sharing the presentation. Okay. Can you see it? Is it okay?
Yes.
Okay.
Barbara is not in the presentation.
Okay, I change. Okay.
Now it's okay.
Okay. So I would like just to briefly present what is EIT Food and which are the opportunities for researcher and for the development under our activities. EIT is the European Institute of Innovation and Technologies, and was born in 2008, and with the aim of strengthening the ability to innovate. Within the EIT, several community KICs have been created on thematic issues like climate, digital, health, raw materials, and of course, food. And every year, new community have been created, and so we are growing year by year. EIT Food, in particular, aimed at make the food system more sustainable, healthy, and trusted, because we focused our activities on the consumer and their perception of the food and of the food system. So we would like to create a trust and fair environment from farm to fork. We are a very wide network because there are, as you can see from the map, member states and also Horizon Europe associated countries within the EIT Food. And we also have a community of over 85 Europe leading agri-food company, research institutes, and university. We have industrial partners, research centers, and university, but also startups, because we promote the growth of the innovation in the system. All the partners. What we would like to do, we would like to face all the food system challenges, because we are going through social, environmental, and entrepreneurial challenges, because population is growing. Over 2 billion people are overweight, while 800 million are undernourished. We have problem with the environment, because about 1/3 of our food is wasted. We need to have global fresh water, and we need that this is not withdrawn. And also we have problem with the entrepreneurship, because about 3.4% of all European startups are in the food industry, and about 9 out of 10 fail because they have a lack of market, or they have not been set up correctly. So our focus is composed by basically 6 strategic objectives. So overcome the low consumer trust. As I said before, we need consumer to be sure on what they eat and what they buy. So we would like to build a consumer-centric connected food system, even with digital systems. We would like to educate, engage, innovate, and advance. So we work a lot with students, PhDs, and researchers in order to improve their capacity to innovate and to work in the food environment. We would like to create a consumer-valued food for earthy nutrition. So we would like consumer and people to be well informed about their nutrition choice and their nutrition value. We would like to enhance the sustainability through a research stewardship, creating promoting the circular bioeconomy, as I was saying at the beginning of this workshop. And we also would like to catalyze the food entrepreneurship and innovation in order to really foster innovation at all the stage of the economic system. AT-Food activities are basically divided in 4 main pillars. Education, so we would like to improve the training of students, but also of professionals by means of summer schools, online courses, but also specific activities focused for researcher and professional. We have we promote the innovation because we with the several events, initiative and challenge labs, hackathons, we bring together all the players. So the young and innovative people with companies, investors, stakeholders, public administration, and putting them together, we really create a network that can promote innovation in the food system. As I was saying before, we promote the entrepreneurship because we really support all the innovative entrepreneurs from the idea to business, with several programs that are divided for just the launch of an idea till the accelerator of a good start up, and also scale up for very consolidated companies or startups or spin-off from university. And then we as I was saying before, consumer is very important for us, so we work a lot with the public engagement, with dialogue events, networking, school programs, but also specific initiative like the consumer engagement labs and focus group, in which we involve consumer and push them to really create something new, like we did last year with Valle Fiorita and our consumer, elder consumer over 65 years old, really create new idea of products, and Valle Fiorita is now launching this new product on the market, very innovative, very surprisingly new. As I was saying before, EIT Food is a consortium of over 85 companies, research centers, and startups. And it's always possible to enter and to cooperate with this network. There are, in the EIT Food status, there are different type of partnership opportunities. It's possible to become a core partners, gold, silver, or SME, a network partner, or an associate partner. Of course, the level of engagement and the level of obligation are different, but you can see everything on the website of EIT Food and check what's could be useful and interesting for you. EIT Food is spread out all over Europe, but you can contact the hub of your personal countries. We are in 17 countries. These are just the one from CLC, Northeast, and the South. But there are other centers, as I said before, the CLC are also in the north of Europe. As per Italy, University of Bari, together with the Impact Hub Siracusa, is the hub for the next 3 years. Here you can find our connection link, so the emails, and you can always write to us for additional information, and we will also send you the news about the event that could be interesting for you. And the aim is really to create the future of food together with innovation, research and development. And if you would like to be part of the community, would like to share idea, and you really would like to help us from the food sector, please be in contact with us. You can find much more information on our website, eitfood.eu. And if you would like to contact us directly, you have our email contacts. Thank you.
All right. Thank you, Barbara.
I was in time.
I think for some miracle we managed to stay at the time frame. Okay. So here we are at the conclusive remarks of today's workshop. There are some facts that we've learned. Indeed, brewer's spent grain is, for the current technology of beer making, is an unavoidable side stream, and it has a great potential as food ingredient. Brewer's spent grain containing food would be considered upcycled food, and this is something quite new on the market. And the question is, what is the future for this type of food containing these unusual ingredients? Well, the answer is below. These are just some examples that I was able to find on the internet about already existing on the market food products. A couple of them contain brewer's spent grain, and others are made with other ingredients. So this tells us about the huge possibility for innovative food products development. What are the takeaway message from today from this workshop? We have seen that proper processing ensures good technological and nutritional quality of the brewer's spent grain. So fermentation and bioprocessing have to be tailored to the type of products that we want to have. We have learned that nutritional value of brewer's spent grain containing food goes beyond the content of fibers and proteins or others. It can also have antioxidant properties, increased digestibility, depending on the type of process that we apply. We have also seen that cereal-based products like bread, pasta, cereals are indeed a great fit for brewer's spent grain, and that's quite natural. It is an easy way to deliver more fiber. They blend well into the attributes of cereal-based foods, and they can increase a diversified, healthy choice for consumers on the market. There are, although, some challenges that have to be faced in the future for really uptaking brewer's spent grain back into the food chain, but with challenge also come opportunities. Indeed, we need to understand and learn what are the processing options after brewer's spent grain is generated. How do we preserve it? How do we pre-treat it for food uses? We have to think what will be the regulatory aspects. How do we store and use brewer's spent grain for food application? There is nothing of the sort, or not available in every country at the moment. We have to really think and invent something about the supply chain. How is brewer's spent grain delivered to their users? What will be the challenge in this respect? Would be possible to imagine a future where the small breweries in the city will deliver directly to the bakers? What are the opportunities in this respect? We have to think also that techno-economical feasibility of different products and processes will be considered and has to be considered if we want to really fully utilize brewer's spent grain also with economical advantages. And we have also learned that consumers' role for this and for upcycled food in general is paramount. We need to engage the audience. We need to start to create and develop more products involving consumers at the most. This is something that the food industry knows very well, and it applies also to this type of food. This is something that I want to share with you briefly, and it is the result of a very recent master thesis studies that has been done at the University of Helsinki. And these are the results of our consumer survey, when we have observed that consumers showed quite some interest in consume brewer's spent grain fortified products in the future, and also thinking about what could be then the different food categories that you would be interested in. Bread, but also snack bars or type of breakfast application were probably the most interesting, but these low numbers still indicate quite a great potential for successful new food development that is indeed possible. And last but definitely not least, I really would like to thank everybody who was involved in the FUNBREW project, the funding bodies, the SAS Food, Eranet, and indeed all the students, researchers, technical personnel, industry, people, steering committee members in every country, volunteers in our panel that contributed to make this project a success. And definitely we would like to thank you today for your participation to the workshop. So thank you very much. I will stop the sharing of the screen, and we are a little bit over our time, but only 1 minute. So yes. I'm very pleased that we managed to conclude in time. If you want to contact us, we are all over the internet. There is our website, and we have an active Twitter webpage. So please feel free to drop an email to talk to us anytime.
Thank you very much, Rossana, and thank you everybody to joined us this morning. It was very, very interesting, and I hope it's just a start for new activities together, as our aim of EIT Food is to create networking. So these are the activities that really fit for our aims. As I said before, all the workshop have been launched live on YouTube, and you can see it at our page, YouTube page of EIT Food Hub Italy. You can then find everything there if you miss something, or you can, if you want to review something that you would like to go deep. So thank again to everybody, and have a nice day, have a nice hot summer.